Good afternoon, everyone. We'll go ahead and get started. My name is David Foley. I'm one of the co-chairs of the Planning Committee, and I want to welcome you to the lunch symposium entitled Providing Access for All, Current Best Practices and Novel Paradigms Ahead for the Highly Sensitized Kidney Transplant Patient, sponsored by Hansa Biopharma. On behalf of AST, I'd like to thank Hansa for their continued support of the CEoT meeting. And at this time, I'd like to invite Stan Jordan to the microphone. He's from Cedars-Sinai, and he's going to start the program.
Okay, thank you very much, and welcome everybody, and to this program where we're going to look at providing access for all. This is going to be a very important theme. I know it's the theme of the CEoT meeting here. And what are the current best practices and novel paradigms ahead for highly HLA sensitized patients? So thanks for coming here. These are the faculty disclosures, and they're available for review. And first of all, I want to introduce our faculty, and we're very happy to have an excellent faculty. First, Carrie Shenstock from Mayo Clinic. I'm sure many of you are familiar with her. And then Oriol Bastard from Barcelona, who will be part of the faculty today as well. So what we want to do today is to describe the changes to the kidney transplant allocation system in the U.S. and their effects on wait list times and transplantation rates in highly HLA sensitized patients. Discuss the challenges of transplanting the highly sensitized patients and the current role of desensitization protocols in improving access of these patients to transplantation. And then provide an overview of the current and potential desensitization therapies that are emerging and their use in the US and Europe. Today's agenda, you can see here it is. We'll talk, first of all, Carrie will talk, then I will give a talk on desensitization therapy, and Oriol will then wrap it up around 1:40. And we'll have questions and answers afterwards. Also, we'd like for you to remember to fill out the program evaluation that's in your packet at the table at the end of the talk. It would be very appreciated. Okay, I think we've already talked about this. This is our esteemed faculty. And without further ado, I'm going to introduce Carrie Shenstock from Mayo Clinic, who's worked very hard on looking at the current state of kidney transplantation in the U.S., especially access for highly sensitized patients to kidney transplantation. Carrie, thank you.
Thank you very much. I'm very thankful to be able to talk to you all today. So as a backdrop, I want to give you some understanding of what the current waiting list looks like. So as of January 2023, there were nearly 90,000 candidates, kidney transplant candidates on the waiting list, okay? Back in 2022, about 26,000 patients were transplanted. That was great. In fact, about 3% increase from 2021. About 80% of these were deceased donors. But nearly 46,000 patients were added to the waiting list in 2022. So there's going to be more and more patients. And of the patients who are active on the waiting list, about 12% of them have a CPRA greater than 80%. And I'll get into more, what does that CPRA mean? And 6% of these patients have a CPRA greater than 98%, or in this more highly sensitized range. So let's talk a little bit more about what is CPRA and what does it mean to be sensitized. So first, how does a patient get sensitized? And when I mean sensitized, I'm talking about they have antibodies towards HLA. Well, the first is just pregnancy. About 30 to 50% of women who've had 3 or more pregnancies become sensitized. Also blood transfusion. Historically, about half of patients who receive multiple transfusions would become sensitized. But today, many patients are actually sensitized because they've had a transplant before. 24 to 90% of patients who've had a prior transplant will become sensitized. Well, how do we measure sensitization, or how do we communicate sensitization? We do that through the CPRA. I know that's not new to this audience at all. And it's just an estimate of the proportion of the deceased donor population to which this particular candidate is going to have some degree of HLA antibodies. And this score goes from 0 up to 100%. And I think you all know no one is really 100% because they don't have sensitization to their own HLA, so it's something that can be up to very close to 100%. A key point I just want to point out is that just because someone has a CPRA of 0 does not mean they're not sensitized. They may still have some immunologic memory if they're not naive. So just keep that in mind. So how do you find this CPRA? In the United States, at least, if you look at your single antigen bead results, you can take the positive results and enter them into this CPRA calculator. It's pretty easy to determine their CPRA. But each transplant center is a little bit different depending on what they use as their threshold of unacceptable antigens. Another point I want to point out is we talk about CPRA, that's the breadth of sensitization, but it doesn't tell us anything about how much antibody patients have. And this was a study that I did with Anat Tambur a couple years ago. It was interesting. We took 20 patients who had a CPRA of 100%, and we did dilutions, serial dilutions on those patients, and then recalculated the CPRA on each of those dilutions. And it was, you know, it really highlighted how different these patients are and really also how much antibody some of these patients actually have. If I orient you to the top of this slide, patient 20, we started diluting the serum and the CPRA would start going down pretty quickly. That person, fairly easy to remove antibody with some traditional therapies, or easier to desensitize. And that's in contrast to this patient 2 at the bottom. We diluted the serum 1 to 16,384, and that patient still had a 91% CPRA. Can you imagine how much antibody some of these patients actually have? So not only do we look at the breadth of sensitization, but also the quantity, or we should think about the quantity of antibody some of these patients have. And highly sensitized patients are harder to match. Again, this isn't news to this group. That's probably why you came to see us today. But just to give you an idea, for someone who has an 80% CPRA, they have to go through about 14 match runs to find a transplant. And this is in contrast to a patient who has a CPRA of a CPRA of 99, where they may need to do 300 match runs. But notice as the precise CPRA increases, you know, these 99.99% CPRAers, it takes multiple 300,000 runs to achieve a compatible donor. Now, historically, before 2014, before they changed the allocation system, you could see there were a lot of disparities in the waning time. People who were sensitized just waned, you know, a median of 5,000 days before they could get a transplant. And this was a major focus of allocation schemes in the early, you know, before 2014. So a lot of this conference, actually the whole conference at large, has been focused on access to transplant, right? And in the sensitized literature, what we think about is how do we maintain equity and also utility? And these are certainly at odds. We have to make sure that there's equal access to people, but if we give equal access to patients who may not benefit, maybe we're losing life years. And so the whole goal of allocation systems is really to balance that. This is a brief history of the kidney allocation in the United States with regards to sensitized patients. And I want to point out some important events. In 2000, actually, this is when the US government published an OPTN final rule that was addressing this idea of equitable allocation for deceased organs. It was only in 2009 where UNOS started to use the CPRA. We talked about the CPRA. But a really pivotal moment for transplanting highly sensitized patients was in 2014. This is when the allocation system changed to really give more access to highly sensitized patients. So what happened then? I'm going to direct you to the red line. The red line was the allocation points that were allotted to sensitized patients prior to 2014. If you had a CPRA up to 80%, you got really no extra points. And those patients with a CPRA greater than 80 got 4 points, all of them, whether you were 80 or you were 100%. And you can see how the allocation system was remarkable because it gave increasing points on this sliding scale for the sensitized patient. This is great. In fact, if you have a CPRA of 99% currently, or after 2014, you get an additional 50 points in regional priority. It's amazing. If you are a CPRA of 100%, you receive 202 points in national priority, and this was to increase access for these sensitized patients. And you can see here the success of this system, right? I'll draw your attention to the orange line. Those were patients in the 98 to 100% category. You can see that there was a dramatic uptick in transplants for that group of patients. And so for most of those patients, it mirrored the rest of the population. But I think the main point, what we've learned, is the allocation system has been great. But unfortunately, there's a group who's kind of still left out, who may not really still have access to transplantation. And these are patients within this 100% category who have this greater than 99.9% CPRA. Unfortunately, they're still not getting transplanted. This is data that we worked on looking at the UNOS list and how the allocation system changed transplantation for this unique group of patients. And if I draw your attention to the blue line, this is adjusted transplantation rates. And then on the x-axis, you can see the CPRA. And what you can definitely see is that as the CPRA approaches 100%, greater than 99%, transplantation rates begin to plummet for that group. In fact, their rate of transplantation is only about 0.6 of the rest of the population. And many of you in the audience may be saying, well, yeah, but that's a really small number. That's not many patients. But actually, of the patients in the 100% category, at this time, most of the patients are in this category now, and about 3,000 patients are on the waiting list as of 2019, have this CPRA in the 99.9% category. So there are these patients, and I don't think it's an allocation problem, it's just that there's not enough organs with unique HLA types to be able to match these patients. And of the patients who do get transplanted, sensitized patients just wait. Some of them wait longer. These are wait times based on CPRA that as of 2018. And you can see of the 100% patients, those greater than 99.9% who were transplanted, more than half of them waited more than 5 years, and over double that of the patients who had a CPRA of 80%. So I think in summary, I want to, kudos to those who have worked on the allocation system. It's been great. It's helped a lot of people, but I hope you come away with recognizing that there's still this disadvantaged group who do not have that access to transplantation. And so and I'm not sure the allocation system can really address this group of patients. So I think there's still an unmet need for this group of patients. So with that, we'll have Dr. Jordan speak.
Thank you, Carrie, and I really want to recognize Carrie for the great work she's done in this area and how important that paper is to show that there are disadvantaged patients who are not benefiting from the current allocation system. And certainly this is something that we want to try to look at in terms of providing equity for those with immunologic disadvantaged situations. So my charge today is to talk about desensitization of highly HLA sensitized patients, and certainly unmet needs and challenges. And this is obviously very, very tough topic and has controversy. And so I think we can hopefully address some of those things in the question and answer session. Okay, am I going on? See. So what are, at least from my standpoint, what are the unmet needs for desensitization in highly sensitized patients? So if we look at, I think we all know that kidney transplantation is the preferred treatment for ESRD compared to long-term dialysis. I don't think too many people would argue with that. However, a subset of these highly sensitized patients with high CPRAs are unlikely to receive offers, as Carrie had indicated. And in my career, at least, we see that these people really don't have well-matched living donors, and they're really dependent on the deceased donor transplants list to receive kidney transplants. And often they're HLA incompatible, we know that. So what I've worked on for many years, and others as well, is trying to come up with methods that would alter this antibody barrier, this immunologic barrier to transplantation to allow us to transplant patients that are highly sensitized. And I think it's, again, I don't think anybody would disagree with the statement that there certainly are no US consensus guidelines that recommend specific protocols, and variable success limits their use. So this is pretty much our situation. Now, just to go back to sort of the very beginning of this issue, and the man that that really brought this into clarity was Dr. Paul Terasaki in this seminal paper published in New England Journal in 1969 when I was a senior in college, where he described the importance of positive cross-match or cytotoxic antibodies to lymphocytes and transplant outcomes. So this was really the dawn of the antibody theory of transplant rejection. And it's the first study to show the importance of HLA antibodies in mediation of severe rejection that later we know was antibody-mediated rejection. And because of this, this led to the implementation of donor cross-matching tests before transplants that really was, for many, many decades, just say no type of format, that if you have a positive cross-match, you cannot get a kidney transplant because of the risk for graft loss. And I think for the decades that I've been in transplantation, going through the late 70s through early 90s, the focus was primarily on T-cell modifications, and rightly so, but sort of the antibody ideas really took back seat to the T-cell mediated rejections, where we thought that would solve all problems. Of course, we know now that that's not the case, that we know that one of the main causes, as I think we'll see in the next slide, well, let me just finish this thought, that if you look at long-term outcomes of kidney transplants, that they've really not changed very much. And around 2010, 11, there was data that became available from evaluations of biopsies in the new Luminex assay showing that chronic antibody rejection was probably the most common cause for graft loss. So, and that this may be why we have poor outcomes. Maybe if we address the B cells and their antibodies, that this would help change that. So if you look at the risk for rejection, we know it's very high among highly HLA sensitized patients. That's sort of a no-brainer. And that people who are allosensitized to HLA, that we know leads to the production of donor-specific antibodies that are HLA-specific. And this increases the risk for antibody-mediated rejection. And so the categories there, I think probably everybody knows this here, but hyperacute, that's Obviously, you put the kidney in and it's blown out right away, where we have antibodies in such quantities that it overwhelms the kidney's ability to protect itself from complement-mediated injury in ADCC. And then acute rejection later, and then, of course, chronic rejection, chronic antibody rejection is one of our most formidable opponents today. And we know that about 30 to 50% of patients have preformed ESAs and positive cross matches before transplant without desensitization. And in January 2023, approximately 12% of patients on the wait list were highly sensitized, and approximately 6% had CPRAs of greater than 98%. So they're not inconsequential. Now, if we look at this, it sort of gives you an idea of how DSAs evolved and how they injure the allograft. And this is an important concept in terms of designing future therapeutics. Early on exposure to alloantigens in the kidney leads to direct presentation of antigens by antigen-presenting cells, to T cells, and these T cells become activated and then can move to germinal centers where they activate the T follicular cells. The T follicular cells then generate B cells that, under the influence of various cytokines and growth factors, become plasmablast and plasma cells, and that make high-affinity antibodies that mediate injury, as you can see on the bottom left, through complement activation, and then antibody-dependent cellular cytotoxicity. And both these processes, and probably others, are important here in mediating injury to the graft. And if we look at this, what does it mean, the donor-specific antibody story? And certainly the Luminex assays have been very helpful for us in this regard. And this is a paper that Alex Lupi did many years ago, looking at a group of, I think, of about 700 patients in Paris. And you can see the top line are DSA negative patients. Their long-term survival out about 7 years is excellent. The thing that's really cool, though, about this is that if you have DSAs that don't activate complement, that's the second line, it's pretty similar. So not all DSAs are going to mediate severe injury. So if they don't have complement activating capacity, and that may be because they're just low titer, they may not have ability to long-term injure the kidney. But those that are complement activating, and one of the things that we've also looked at in our lab we won't talk about today is ADCC. We can look at ADCC and how much gamma interferon is produced and how much killing is produced when an antibody binds, or NK cells bind to these antibodies. Those are markers for very poor outcomes. And then this paper, I think, on the right you can see is sort of comes to the same conclusions. One of the things that we did at Cedars a few years ago, and this is Mark Haas did this paper, a very nice paper, I think, that we looked at the difference in pathological features of antibody rejection and what would predict poor outcomes from comparing the patients who are transplanted as highly sensitized patients and those who develop de novo DSAs after transplant. And if you look at this, the graph, that if you look at DSA patients at transplant that are positive, they actually do better because we know what we're dealing with. We can see a positive cross-match. Here, this group of patients really would respond to current therapies like IVIG, plasmapheresis, Rituxan, that sort of thing. And we can flip them around. But those who develop de novo DSAs, and if you look at the pathology on the right side, my far right side, is that you see two types. Type 1 would be acute antibody injury, and then type 2 is chronic antibody injury with transplant glomerulopathy. Those patients do not do very well. And one of the things we found in the study that I think is very important here is that the survival of these patients' kidneys really depended, and when we gave them treatments, depended more on one thing than anything else, and that's our ability to get rid of the antibody. If you can see that the antibody reduction scores were good, the top line, you have very good survival out to 60 months. If we couldn't change it, they just went down the hill. So reducing antibody is critical to solving this problem. And again, if we look at the data from France and then other centers, and in the US, there's about 40% to 50% of kidney transplants who lose their kidney do so because of chronic antibody rejection. So it's a major cause. And this was a revelation a few years ago, and it's become really something we have to deal with. And we know these people, if they lose their kidneys, they come back to the transplant list, and they're very highly sensitized and much more difficult to transplant. And this is an important paper that was done several years ago. I think this is by Bob Montgomery's group and was in New England, where we looked at, or where they looked at, and I think we contributed to this as well, living donor patients and look at the patient survival over time and compared to people who remained on dialysis or those that were on dialysis and got transplanted without any desensitization. You can see here overall for both survival benefit that patients that received desensitization had a life-saving benefit from transplantation, even if they had very high HLA antibodies. So if we look at access to transplantation for highly sensitized candidates, desensitization needs to address improved access, facilitate transplantation while balancing the risk for antibody rejection, and provide highly sensitized candidates a greater survival advantage over remaining on dialysis. Certainly our goal is to reduce or eliminate the DSAs, allowing a negative cross-match and safe transplantation. And the treatment protocols exist for this. However, none are FDA approved, as we mentioned. And this is there are paradigms out there, but one of the things that we would look at is that we have, most important, if we do have living donors available that are without antibody, certainly that's the one you want to take. If they have DSAs, then you want to try to look at paired donation. But for many of the very, very highly sensitized patients, this is not an option because people remain on these lists for long periods of time. And from my standpoint, they have a better advantage on the deceased donor list because they have a national sharing at this time anyway. So again, if we don't have living donors, we go to the deceased donor, and then we have our paradigms for treating them and trying to get them transplanted. Therapies for desensitization, pretty much three things since the early days: retux, plasmapheresis, or low-dose IVIG, and high-dose IVIG with plasmapheresis. So these protocols and timing differ and with variable access, but I think across the United States and probably the world, this is what is being done for most patients at this time, or variations of this. And this is a bit busy, but it talks about the plasmapheresis, what it does. I think you know about the mechanism of actions, adverse effects. Certainly we worry about removing coagulation factors with bleeding. IVIG, high dose can cause some side effects, but we give it on dialysis usually and see much less side effects. Anti-CD20s are also important to prevent rebound memory responses, at least in our experience, and putting these together has worked pretty well, but still not all the time. And so certainly current desensitization methods may limit utilization. Timing of desensitization varies, and it's very tough to do because you don't know how long these patients are going to wait before they get a kidney offer. If you give it at the wrong time, you may miss an opportunity, so you may have to repeat it. So all these things are difficult in terms of knowing how to do it when you don't have a living donor. So in summary, although the risk of antibody rejection is high among highly sensitized patients, candidates, desensitization can increase access and improve transplant outcomes for this underserved population. There's no current consensus guidelines that exist in the US for desensitization, and protocols vary. by institution, typically requiring planning and a lot of resources. The need for development of desensitization protocols to improve outcomes and timing for desensitization is ongoing. Okay, thank you very much. And now I'm going to introduce Dr. Oriol Bastard, who will talk to us about potential methods for transplantation in the unmatchable, highly sensitized kidney transplantation. Thank you.
Hi, good afternoon. Thank you very much. I'd like to thank the organizers, Hansa, for the very kind invitation to come all the way from Europe. And you will see my talk will basically try to give you a picture of how in Europe are we trying to face this problem of the highly sensitized patients. And actually we tried to organize this and was endorsed by the European Society of Organ Transplantation. We made a sort of a guideline, try to put, let's say, the messages that we wanted to clarify when we talk about highly sensitized patients. So I think this figure, it just illustrates what Carrie just showed, but only in the US. And this just wants to illustrate the number of patients that currently we have in worldwide and are considered as highly sensitized. As you can see, it depends more or less on how we define sensitization, what are the assays, most of them by using solid phase assays. But you can see that in between 10 to 15, even to 20% of the patients on the waiting list are considered as highly sensitized. And I think, and actually Dr. Jordan just showed this slide, we all have these figures in mind, and we know that transplanting across the HLA barrier is deleterious for the graft, and this was shown, as you can see, both in a US trial, a retrospective trial by Bob Montgomery's group, where you can see very clearly the differences according to the assay that you're using to define a highly sensitized patient, whether you just use solid phase assays, whether you have a physical cross-match or not. And this was exactly replicated in Europe, in this case in the UK, so showing indeed that the results of transplanting these patients as compared to an HLA compatible is significantly worse. And I think, however, and this is something that we are struggling somehow, how we can consider and compare these patients when we do transplant them. And probably, and as Dr. Jordan also mentioned in his presentation, the comparison of these patients should not perhaps be with patients that receive an HLA-compatible transplant, but conversely to patients that are remaining on the waiting list, and they, as you can see, they die on the waiting list. This was nicely shown in the US. However, in Europe, perhaps because of different healthcare systems or wherever, this was not really the same case, but was proof of regarding quality of life and so on. So I think this is something very important, that these patients probably should not be compared to an HLA-compatible transplant, but rather if they remain forever or for a very long period of times in the waiting list. So this is somewhat what we try to put together in this consensus, which was called the Engage, to try to force or to foster the transplant community to transplant our patient with an HLA-compatible donor. And these are three main programs that are developed or implemented in different countries in Europe. Basically, we have the priority points in deceased donor kidney allocation program, which is very similar to your KAS program. Then we also have the living donor kidney pair exchange program, which is a little bit diverse in each country, but it's also implemented. And also we have the acceptable mismatch program, which is also implemented in some countries, and particularly, as you know, in the Eurotransplant area, which is also being performed to try to booster or to find HLA-compatible transplant in these patients. And this is just a figure that tries to summarize these three programs and the outcomes. And in the left panel, you can see the PATHI program, which is the KAS program, which is performed or developed in Spain. And you can see how by prioritization in this sliding scale score system, we could also offer an HLA-compatible transplant to this very highly sensitized patient, to those showing a CPRA on the 98% or also in the 99%, but you can still see that the 100% CPRA were still remaining despite being prioritized in this program. Then you can see also, I think this is coming from here, from the US, how the implementation of this living donor kidney paired exchange transplant did reduce inversely the number of desensitization programs in the US, so suggesting that all these patients were receiving an HLA compatible rather than going or undergoing a desensitization program and receiving an HLA incompatible transplant. And finally, you can see also the acceptable mismatch program, which is kind of very similar to the prioritization program, the KAS or the PATHI program, but trying to change the concept and extend somehow the HLA type of the recipient to the donor. And you can see again that those highly sensitized below the 99.9% are also capable to find a compatible organ. However, this is not the full picture, and you can see when you focus only the CPRA what's happening to these patients. And for example, in this prioritization program, you can see that they don't really receive not a transplant, but actually no offers for an HLA-compatible transplant after being at least 1 or 2 to 3 years in the list. And the same occurs when you look on the living donor kidney paired exchange. We know that after 5, 6, or 7 rounds of trying to find this compatibility, you can see that you or these patients will never find also a match HLA-compatible transplant. And this is actually occurs exactly the same with the acceptable mismatch program. So we have a pool of individuals, these very highly sensitized, above the 99.9 or 100%, that remain on the waiting list. So this is a little bit the figure, and so while we're capable of transplanting highly sensitized individuals, we still have an important number of population that is actually increasing over time and that are remaining on the waiting list. And also the question that is raised and that we all pose to ourselves is who can undergo a desensitization program in a, let's say, safely manner. So this is also what we kind of put together as a conclusion, and based on evidence-based reports in the literature, what is being done across the globe, in the US, and also especially in Europe, as you can see, and as Dr. Jordan showed, is basically based on the literature and old trials done using plasma exchange or immunoadsorption nowadays, and also targeting B cells either by using IVIG, high doses IVIG, or anti-CD20 monoclonal antibodies to target or deplete B cells. But however, I think there is also, and as you all know, we are in a setting, a kind of an optimistic situation, that there are a number of molecules that are on their way being evaluated in different trials that I will just briefly summarize in my talk. I think in this cartoon, I think it's also, and Dr. Jordan just mentioned that, I think it's very important to understand how we classify highly sensitized patients. And this just wants to show how do we describe or classify particularly serological memory, which is what we're using in our daily clinical practice by using different assays. But as you can see that the outcomes of being transplanted across a virtual cross-match, but with a negative physical cross-match, or by being transplanted with a positive flow or a CDC cross-match, it really differs in terms of outcomes, in terms of the risk of developing hyperacute rejection, or a short or mid-term chronic antibody-mediated rejection. So I think this is very important to bear in mind when we want to plan a desensitization for one of these patients. So what do we have, or what we have been doing when we're trying to implement this desensitization program? So I think this cartoon also illustrates in a very simple manner the intricate network of the humoral immune response by showing different compartments where all these cell subsets are playing a specific role finally or ultimate to produce this anti-HLA or donor-specific antibodies. So we've been trying to target terminal center reactions, so B cells or TFH cells. We've been also trying to target bone marrow residing long-lived plasma cells or B cell residing in the bone marrow. Also in the bloodstream, trying to get rid of these circulating antibodies and also B cells, and also finally to protect the endothelium by using some complement inhibitors. And these are a little bit the molecules that we do have or that are actually in the way of being evaluated for their efficacy, for the sensitization. But as you know, there is no, at least here in the US, no FDA-approved drugs for desensitization. I just wanted to summarize here those molecules that are particularly targeting a specific compartment or cell subset, the B cells, anti-CD20, IL-6 blockade, the anti-Bleeds. Then we have the proteasome inhibitors drugs. Also, we're now understanding how the anti-CD38 monoclonal antibodies against plasma cells are working. We have the IgG degrading endopeptidase, as you will see, and we have complement inhibitors, and also we are trying to understand whether by using co-stimulation blockade we may block or abrogate a humoral immune response. So very briefly, trying to target B cells, there is this third generation of anti-CD20 monoclonal antibody, the obinutuzumab, which has shown actually to be highly effective to deplete peripheral and also lymph node B cells, but has not still demonstrated that it's capable to really reduce or downregulate the amount of MFI antibodies or CPRA. Regarding the anti-IL-6 blockade, we have two different molecules. One is just targeting the receptor of IL-6, which is tocilizumab, and has shown certain efficacy in treating antibody-mediated rejection. And it's associated, it has shown an association of kind of a reduction, a slight reduction or DSA scores or MFI values of DSA. But particularly in the setting of rescue therapy for AMR. The other one targeting directly the soluble IL-6, clazakizumab, is a pretty interesting one. Also has been shown certainly some efficacy for rescue therapy, and is being actually evaluated for chronic antibody-mediated rejection. Has shown in some retrospective or single-center study that is capable also to somehow reduce the levels of donor-specific antibodies also over time if it's treated chronically over time. The proteasome inhibitors, you all know them. I think bortezomib was the first one, as you know, that was developed for in transplantation or implemented in transplantation, and showed a very effective value in terms of depleting bone marrow residing in plasma cells. However, you know that it's associated with a substantial neurologic and hematologic toxicity, which made that it was changed or substituted with a second generation irreversible proteasome inhibitor, the carfilzomib, which is actually working in the same way, but has shown significantly less toxic effects as compared to bortezomib. I think interestingly, as you will show later on, I think now it's being tried to use in a dual targeting manner, both in the periphery by co-stimulation, using co-stimulation blockade, but also centrally in the bone marrow using this drug. Then we have these two new drugs, these monoclonal antibodies targeting CD38 surface marker, which is expressed in mature B cells or plasma cells. And actually there is a trial which is probably, the results will be published very, very soon, and in the ITC there will be some of the results which are kind of promising, showing how do they deplete these cells in patients that are highly sensitized. And then also we have the imlifidase, as you know, is an endopeptidase derived from the Streptococcus pyogenes, and as you know, it has this unique function that cleaves both soluble and also the BCR, the IgG, which is in the surface, the B cell surface from the FC fragment, and it abrogates simply the function of both the FAP and also the FC part of these antibodies. The administration takes, as you know, it must be done within the 24 hours, and it has 5 days of efficacy in terms of depletion of all these soluble donor-specific or anti-HLA IgG antibodies. And finally, just to conclude, I think one of what we have been learning over the years is that perhaps not only one drug should be used for this very highly sensitized, but rather to target different compartments. And as you can see here, there are different trials ongoing. For example, blocking B cells or TFH with co-stimulation blockade with belatacem at the same time, and using anti-CD38 monoclonal antibodies with daratumumab. There are 2 trials ongoing. Also using a proteasome inhibitor, carfilzomib, with also a co-stimulation blockade, and also using an anti-CD40 ligand with an anti-CD19. So in other words, so trying to block different compartments at the same time might be perhaps more effective than just using one sole drug. And just to summarize, once we want to make the decision to go or to not perform desensitization, I think there are many, many variables that we have to bear in mind or take into account. One are the immunological characteristics of these patients. We can decide whether to delete, for example, antibodies or not that are directed to previous antigens, so to avoid memory B cells or recall responses. The amount of MFI, which has been linked to the complement capacity of fixing of these antibodies, and whether also by using titration, as Kerry just showed, can help us to decide whether there are certain antibodies that we can avoid or delete to move on to desensitization and to transplantation. But also, and this is probably the most difficult thing, is we have to deal with the patients, and normally these patients are not the easy ones to do. And I think this is something that we have to really bear in mind, because our patients that are going to probably receive high amounts of immunosuppression, so frailty patients probably are not the best ones or the best candidates to put in these programs. And also at the same time, take into account the donor characteristics. Probably we want to avoid long cold ischemia times, avoid DGF, to try to avoid immunogenicity. So there are a number of both immunological variables, but also recipient and donor characteristics that we will have to bear in mind. So just to summarize my talk, there are strategies and programs that are currently in place in Europe to try to maximize as the first option an HLA-compatible kidney transplantation. There are numerous emerging therapies which are being currently evaluated for the sensitization and also treatment of AMR, both acute, subclinical, clinical, and chronic AMR. A depletion of DSA immediately prior to transplantation as well, to concern for immunological memory, and particularly the control in the long term of these antibodies is something that we still have to understand, and probably we need to understand much better the mechanisms of the humoral immune response. And that finally, the therapeutic strategies are under development, but long-term safety and efficacy need to be established. And I thank you very much for your attention.
Thank you, Oriol, for that great overview of the emerging therapies. In terms of closing remarks, I think that I feel very privileged to be here today and to discuss all the aspects of desensitization that we went over, and really trying to find a place for this therapy for these underprivileged, immunologically disadvantaged patients, trying to get a kidney transplant to improve their lives and the lives of their family. And I think that, as in concert with the theme of this conference, is that we all have to look at ourselves as custodians of the kidney allocation. We try to make the best choices for all the patients that we deal with. And certainly, from my standpoint, most of my career has been focused on the immunologically disadvantaged HLA, highly sensitized patients. And And it is really encouraging to me to see all these new developments, and that there are now many different biotech companies interested in developing agents for use in transplantation, not only for desensitization, but for treatment of antibody rejection. And I think we should always try to make this in our own hearts, our own minds, that we want to provide access for all, and look at what are the best options for all our patients here, and how who would benefit from desensitization and who might not. And certainly having new drugs that could open doors for these patients will help, I think, in those decisions. So I think we're on the verge of a new era in transplant medicine, and I think will be very interesting to see where we go in the next few years with these clinical trials of these antibody-modifying agents. I'm sure Dr. Terasaki would be very proud of the work that's going on right now. So thank you all for your attention today, and we're going to open it up for questions now.
Well, this was great. I'm Göran Klintman, Emeritus Chief from Baylor University Medical Center and involved in transplantation since 1978, kidneys and livers, etc. This is a very interesting issue that is being presented today and discussed. And what was brought up here early on today in this discussion was these patients who are waiting for a long time, can we give them priority to get them transplanted? And I think that that is a valid question. And then how do we get them transplanted, and how can we actually provide immunosuppression for them so they actually can retain the transplant? Mm-hmm. Well, that is the topic today, but I want to bring up, as I always do, the other side of this coin. If we take these highly sensitized patients, treat them with novel therapy, what is the graft survival? And if the graft survival, and I'm just throwing it out, say that the graft survival is 60% at 3 years, can we justify using kidneys for a patient that will only keep the graft for three years? Can we justify that when we have other patients who may benefit from that graft for the next 20 years? And I think this is a critical question that needs to be brought up when we talk about allocation schemes.
Okay. Thank you.
Thanks. So I think the slide that you put together showing the immunologic risk is really critical there. So when you're thinking about these patients, it's not that you're going to do everything. You have to be very calculated and think about what are the risks that we're going to take, okay? So, you know, avoiding the maybe CDC positive cross matches, try to get something that's a little lower. Our experience at Mayo, where we did the positive cross match patients with high flow positive, negative CDC, about 75% of those grafts were still working at 7 years, which, you know, I think is, you know, pretty good. So you have to find that sweet spot, and I think that's maybe what you're getting at. It's not for everybody. It's not you do everything, but you find out what is going to, you know, balance that.
I agree with that. I'm just saying, this is the issue when we're talking about allocation schemes, and how do we justify throwing kidneys to patients who have a limited graft survival and leaving patients who would benefit for 20 years still on the list? And that's the question.
I'd just like to comment briefly on that. I think that from my standpoint, that, I don't know if this is on, Is this, can you hear me? Okay, I got it. Yeah, good. I've got it. Okay, thank you. Yeah, so the question is here, if this is the case, and and this is my opinion, I think if we're afraid of the immunologic barrier, then we should not give these people extra points. You should put them at the bottom of the list and say you just have to survive on dialysis. I don't think that's the kind of people we are. I think that we're looking for innovative technologies, and there's no question we have to be held accountable for outcomes. And if we can put together these kinds of protocols, then I think we should expect these kidneys to last as long as patients who are not sensitized. So that would be my feeling about it. You have a question?
Yeah, I just going to thanks for the presentation. So I have a counterpoint to Dr. Klipbaum, just something to think about, and then just a question or suggestion. So, you know, the one thing to think about the people that do, let's say, hopefully are transplanted with this, you know, with the sensitization therapy, you know, I think we'd be incumbent upon that there's a lot of structure for them post-transplant, right, that many patients don't do. So you put them in a position to win, you know, meaning that, you know, the role of exercise, all the rehabilitation aspects. So maybe that's something that could be part of this, and maybe set as a good example for the rest of the community that's not being done. So I'll err on the side of patient advocacy there. But the other thing I would just say too is that looking at these different clinical trials, and this group knows this better than I do, but unless they have a Stan Jordan or Dr. Shintok, these patients are lost. So how do you make it so that when these clinical trials come out, it's not just up to the fact that I've got a doctor who's aggressive with clinical trials, right? So that from my standpoint, it almost seems like, and again, not trying to add another thing on your list, but maybe this is something to do with the other patient organizations, is to take a look at how do you build a community for these patients so that they're empowered, right? And so these clinical trials, and you give them hope. So anyway, just a thought or suggestion.
Thank you. Other questions? Yes.
Thanks. Ben Vernon from Denver, Colorado. This has been fantastic. And if you don't mind, I have several questions. The first is there a way, treatment for acute antibody-mediated rejection, is there anything that we can learn there where we have a greater body of patients to work with, if you will? What can be learned there that can be translated to desensitization?
Yeah.
Yep. Well, I think the setting is completely different. I think acute AMR, it's pretty, so it's difficult, of course, event, but I think we can kind of have weapons to control them, unlike chronic AMR. I think it's these acute AMR that occur in the very first weeks after transplant. I think we have pretty good tools to treat. Treating the sensitization, particularly in those that I think we all are referring to, which are these really highly sensitized, 100%, I think this is a completely different story. And that's why I just showed this cartoon about the different compartments that are completely activated, and the network is perfect. So, and I think translating these drugs in a patient that has everything activated is completely different, and for ages. So I think it's a different setting.
I think everybody in the room appreciates that transplantation is not like love. It's not better to have transplanted and lost. One thing we learned from the kidney allocation transition, the patients we got transplanted, at least in our institutions and what I can see, were full house mismatches. I mean, they were 6 antigen mismatches. And with a panel as distinguished as this, why? We just happened to find the right key for the lock? Is that what the system did?
Well, I think that's a pretty deep question. I think that there, you know, as Carrie, go ahead and take this.
I'm not sure if I'm getting the question. Are you saying that most of those patients were not matched who got transplanted?
Yeah, I think if you look carefully at the data, you'll see that there were full house mismatches. I mean, there were 6 antigen mismatches. They just happened to fit the lock, I think.
Yeah, I think if you look at the 100%ers who were transplanted, a lot, I mean, there was a higher proportion of 0 mismatches in that group. But certainly there's that 99.5% who are 100%ers. They got so much priority, but there was organs available for them. So that may have been just the ones that they got. So what we're really referring to in this talk is these people in the 99.9. The 99.5, I think the allocation system did those benefit. They got transplanted. Those people get transplanted very fast. It's just this group, there's just not enough organs. I hope I'm answering the question.
My last question is, with this robust panel, with 50 years of retrospect, what was the mechanism for the transfusion effect?
Yes, I'd like to take that on. I had some grants on that back in the early 1980s with the NIH, looking at our work was looking at you know, some of the work that came from Dr. Terasaki's lab suggested that the more blood transfusion you got, you actually did better. And we thought that there was some immunomodulatory effect, and we looked at anti-idiotypic antibodies. We found them in pregnant women and then in some of the transplant patients that were blocking HLA. This was our thesis for this grant, but and we were doing, as you know, in the early 80s and late 70s, DSTs, or donor-specific transfusions, to improve outcomes in living donors. Of course, that all stopped with that funky HIV virus that emerged, and then the advent of Epigen reduced the need for blood transfusion. So from my standpoint, and we were talking about dreams and stuff like that earlier in our discussions, I was thinking, my career's over with this anti-idiotypic stuff, but then I'm a pediatrician by training, and we used a lot of IVIG, and I thought, my God, one night I woke up and I go, well, that's a lot of antibodies, it's not infected. Maybe those antibodies are there, and we did work showing that they indeed were blocking HLA in vitro and then in vivo. So that's how the IVIG story emerged from our standpoint. But I think that the blood transfusion effect, nobody really ever knew whether it was inducing blocking antibodies. We definitely did see some people do really well with it, but that was all before cyclosporin. And after that, it just sort of disappeared. Yeah. Yes, John?
Yeah, hi, Kobashi, Los Angeles. Question about, you know, we've tried in cardiac transplantation, we've tried many of your therapies, Stan, and tocilizumab, daratumumab, and now, you know, we're going on to co-stimulation blockade. And I saw the trials that are up there. Are they meant for treating antibodies, high levels, or prevention post-transplant of DSA? How do you see the role of co-stimulation blockade?
Yeah, I think that's a really good question. So the two trials are meant to be evaluated for desensitization, both of them. One is with daratumumab, and another one is with a proteasome inhibitor. And they are both meant to try to block the peripheral memory B cells and TFH, which the CD38 does not block, the immature ones, and combine them with the long-lived plasma cells and plasmablasts. So it's basically meant to try to see whether the CPRA can be downregulated or reduced to have, you know, this patient get some offers by reducing the MFIs and the antibodies. But I think you raise a very good question, is what can we do afterwards? Because we are doing very fancy and or evaluating new drugs, but then we put them in the same triple therapy, right? that we know is not really probably maybe the best for these particular patients. And I think here, for example, continuing with co-stimulation blockade or evaluating their value would be very, very, very valuable. The problem is how you we've been discussing this a lot. How can you design a trial with these patients that you have achieved, you have avoid hyperacute rejection, you have avoid acute rejection to try to see whether by changing to a co-stimulation blockade is better or not, which probably is likely according to the mechanism. So I think there is a lot of work to do here to better understand. Yeah.
I'd like to just comment on that too, because we did a study, a center-specific study at Cedars with clazakizumab for desensitization, and we found that it worked very well with Plex, IVIG, followed by monthly sub-Q injections. They were able to transplant 20 of 20 patients, and about 60% of these were 100%ers. But what I think was, for me, the idea we had, and actually was very revealing, is that we continued it after transplantation as part of their standard protocol with reducing MMF. And we saw really no rebound. And over time, at 6 months to 12 months post-transplant, we saw big increases in Tregs and regulatory B cells coming up. So some of these drugs may have benefit in terms of altering the immune environment, changing it to more of a regulatory pathway than inflammatory, where we're sitting there, and also control these rebound phenomena. So I think this is really, your point is very well taken, that these drugs may be very important to continue after transplant in some way to prevent the rebound phenomena and the slow destruction or chronic antibody rejection that might come from that. Other questions? Okay, if not, thank you everybody for coming. We really enjoyed the program. Thank you. And thanks to our great speakers here.